Okay, I guess we can start. Good evening, everyone. I would like to welcome Professor Angie Serrano from Boston University. Dr. Serrano actually holds the largest KAT6A and KAT6B biobank. So she stores blood for KAT6A and KAT6B patients and also has iPSC, and she will talk more about what iPSC means to us and for research.
Thank you so much, Natasha. I thought about, first of all, thank you everyone joining now, and even a bigger thank you to those that are willing to watch this recording afterwards without a chance to asking questions. I will set up a Q&A session, so if there is any question remaining after this session, I can give the opportunity to everyone to learn about what we're doing. I think it's super important that you know exactly what happens when we ask you to participate in the studies. And in this particular situation, I also want to share with you the big picture of our lab and what we do from the basic science perspective, and why do we think that this matters a lot in terms of advancing what we know about rare disorders like KAT6A and KAT6B. So I'm going to for myself a few slides for a small introduction, and please feel free to interrupt at any stage. This is not to be a monologue. I would love for questions if you have any. So in our lab, we do what is known as basic science. We basically ask questions about how things happen. The concept that we use to ask those questions is during development. When a baby is developing in a mother's womb, we want to know how are the decision-making steps that each of the cells in an organism are taking towards becoming a brain cell or a gut cell or a heart cell. And proteins like KAT6A, KAT6B, and the other proteins that we also studied, like KMT2D, KDM6A, are super important to allow each of those cells to access the genetic information at the right moment to know what they will be and how to become those different cell types that form a whole human body. For that reason, here I'm just putting an example. Probably many of you, whenever somebody Googles KAT6A, KAT6B, what do they do? And one of the common languages that you will find is that they add acetyl groups into the histone tails. So what does that mean? It means that all the genetic information in these cartoons here are these greenish lines. All the genetic information here, it's packed in a unit that allow us to fit into a cell. But to be able to read that information, we need to let the cells unpack that information and pack it again whenever it's not needed in that moment. So what these proteins do is actually control when this information is accessible. And there are many different marks that this histone acetyltransferase or methyltransferase is used in order to make that information available. In this case, I'm just putting a few of them that we study here in our lab, but then I will go to the specific case of KAT6A and KAT6B. So as I was sharing with you, this decision from being a cell that knows it's going to be a brain cell, but it doesn't know which kind of brain cell it's going to become. All this process occurs in a timed manner during development. And in our lab, we focus specifically in the processes that originate a brain and a cardiovascular system, which means the heart and also the main vessels that go into the heart. And then I was just sharing with all of you how important it is to have this information available. So what happens when we don't have what we need to know or to access that information, like, for example, the KAT6A, KAT6B, or other histone modifiers that allow access to this genetic information. So, and in this cartoon over here, there are three different situations. In the first one is we have all the amount of protein that we need. There is no mutation, no pathogenic variant, so we have all the protein necessary to access that information whenever it's needed. Some other situations in which KAT6 disorders also fall into is that we have half of what we need to access that information, and to this day we still don't know what that means for the development of a baby in the mother's womb. Which is one of the most important questions that we need to tackle. And I think that tackling from the basic question, what does it mean to have half of what you need to find your identity as a cell, to become a brain cell, a gut cell, and then to interact between them, I think it's something that needs to be answered. And then this is not a situation that happens with KAT6 disorders, but in some cases, you don't have that protein at all. In our lab, we do use this model in which we remove the protein completely from a cell just to have the extremes. This is what happens when you have it. This is what happens when you don't have it. And then we go in between and ask the question, what happens when you have half of that information? So these are the three scenarios that are possible, and most generally speaking, and we study rare disorders of epigenetic origin in these three cases. So I keep talking about rare. I think that now most of you are actually know more about what this means for in your life and with your family and with your loved ones. Sadly, from a social standpoint, rare means that there's not enough patients to either start the clinical trial. There's not enough patients to attract the interest of company to develop, to understand what it means to have these pathogenic variants, and also to discover or develop drugs and treatments for these rare disorders, because it affects very few patients. And in the US, that means that it affects than 200 individuals. But what we want to do right now is to take this from a different perspective. We grab all these disorders that have a common origin. For example, we have pathogenic variants or mutations in these histone modifiers. So it's not only KAT6A, it's not only KAT6B, it's also KMT2D, it's also KDM6A, and some others that have a similar disease mechanisms. So then it's not that we have just 1 in 200,000 individuals. When we take all these rare disorders together, it's like 1 in 10 Americans actually have some kind of rare disorders. So if we picture all of us going to an elevator that can fit 10 people, with these numbers, with these figures, one of those people will likely have a rare disorder. So this has a different impact, and this is the kind of, as well we say, if we come together, we are stronger. And I think that this is one of the approaches that we should consider as a community of rare disorders, and it's definitely the approach that we have in our lab. So now what we want to know are some of these disease mechanisms shared? Are they regulated in a similar manner during the development of a baby? And I think that having patient-derived iPSCs model, which we're going to go into in a little bit, is going to help us to answer some of these questions. So I've been talking about making this information available, so I thought that we could probably have a bit more of in a cartoon about what I was sharing with you. So I put some of the language that I think that most of us find when we Google this, which is the first thing everybody, even as researchers, when we don't know something, the first thing we do is just to go and search online what we have. So this is one of those things. Histone methyl and acetyl transferases, transferases, sorry, that's my husband calling me. 1 second. Are also known as writers or readers. What does this mean? Writer means that we have these enzymes or these proteins adding something into the histone. So that's why they are known as writer. And they can add either methyl or acetyl groups. In the case of the KAT6A, A and B is acetyl groups. And this happens differently. It happens in a specific manner, depending on what cell type we have, and also depending on the time that this is happening during the development. So all of this, what basically does in one cell type is to allow for specific information to be accessible. That information translates into the elements that the cell needs to either become a specific cells or to respond to the environment. And when we have a pathogenic variance in those or mutation in these proteins that access or allow the access to this information, this happens differently in the information that is accessed is different depending the cell type, or also during the moment of development. So then the clinical features of what we observe in children or in patients, it's actually different. And we know that these kind of disorders are known as syndromic because it actually affects several organs and systems. So this is basically what I was trying to represent with these cartoons, that while we have a pathogenic variant in one gene, all the amount of things that get changed or that get in a different kind of situation at the end, it's not known exactly, and it does differ from patient to patient, while there are some common things that we are going to discuss in a second. So we basically do not know exactly how all of these marks that allow the access to information are altered. While we know that KAT6A and KAT6B have acetylase-specific parts of these histones, we don't know how this affects the general landscape of that cell. Are only these marks affected, or are other things that are affected in this system that allows for access to information? We also know very little about what happens when we have an alteration in how this information is accessible, and we know even less about what does that mean for a child. So I think that's why we studied this from the perspective of basic science, developmental biology, and what it means to have these pathogenic variants. So going back specifically to the case of KAT6A and KAT6B, this is, again, the same thing in a different shape and color. In yellow, we have the DNA or the genetic information. KAT6A and KAT6B allows for that information to be accessible by adding these acetyl groups into this system. And when that happens, and while we have two different genes, there are some common traits that are associated with pathogenic variants in KAT6A and B disorders. And I just took information that you are already familiarized with, which is a very well put together handbook from the foundation, in which you can see all these common traits or clinical manifestations that children have that are common to both KAT6A and KAT6B. And as you can see here, intellectual disability, global developmental delay, congenital heart defects are amongst the common traits. And this aligns together with what happened with other rare disorders of epigenetic origin, like KMT2D-driven disorders that are also known like Kabuki syndrome. It's a completely different disorder, but it does share some of these common traits. And that's why we study them, at least in our lab together. So before going to the KAT6 bank research in progress and just the status of our bank, and just to walk you through what this means, I wanted to know if you have any questions so far up to now. Okay, so I didn't prepare too much about in general about what it means iPSCs in the slides, because there are other recorded talks from the lab in general, or even my talks in the past, in which we walk through what it means an iPSC. But I will super quickly go over what an iPSC is and why it's so important to have a bank that is open biology, which means that it's accessible to everyone who requests these cells. So one of the greatest advances that we've done in research is to be able to grab blood cells, or even a skin punch, or where we have cells that are known as fibroblasts, and then ask them to forget everything they know about their identity and to become an embryo-like cell. Those are known as induced pluripotent stem cells. Induced because we grab cells that are mature, that they are part of your blood, and we make them this embryo, we push them to this embryo-like status. And then pluripotents because they can literally become, if we give them what they need, a muscle cell, a brain cell, a gut cell, a lung cell. And here at the Center for Regenerative Medicine, where I have my lab, we have different research labs studying these cells in terms of making them liver cells, lung cells, or stomach cells. And in our case, we do heart, blood vessels, and brain cells. So this is something wonderful because now it means that we can have a patient cell from just from blood or from a skin punch, and then from that one sample, have the ability to study many other tissues and cell types. And this is not as perfect as some people would like it to be, like we're ready for cell therapy for most of these tissues or cell types, but it's as good that right now there are many studies in clinical trial and are being actually used to recover or to ameliorate some of the symptoms of very devastating disorders. So it's something that we do fiercely here at the CREM, and we think that this is a good and robust path to understand how can we have a snapshot into development while of what are the processes that happen during development in patients without the need of having invasive procedures. So once you give us blood or donate the blood to the bank, we grab those cells, we run them through specific research protocols, and we store them. And once we have that, this is literally limitless source of cells, because we can make them divide and generate more and more cells, and we store them. So once the cells is an iPSC cell, it's banked, and we can distribute it around the globe without giving up the main source of the cells here in the lab. So there is no limit to how many cells we can share. So after that very quick introduction, but I will be more than happy to answer any questions about that. I just wanted to share with you what we've been doing with the samples that many of you shared or donated to this bank. So for us, it's very important that we can represent the whole population of patients. The reason why we do this is because there's so little that we know about these rare disorders that we want to generate a resource that will stand up to any scientific discovery, that will actually be a resource for everyone and every researcher without a specific research hypothesis in mind. Let's say some groups are studying one specific kind of variant, some other groups are studying other kinds of variant. We just store the whole universe of diversity that we have in our patients, and then as we can, and in agreement with the KAT6 Foundation and the research collaborative network of the foundation, we try to establish an order of priority that will most likely benefit most of the patient population. So it's not like we assign priorities without discussing with all the research community. We just are as mindful as we can to use the resources that we have to better serve the whole patient community. So as I was saying, it's important for us to have both variants well represented in our bank. So we collect blood from patients KAT6A and KAT6B, and also we pay attention into the age group and also the sex and even the racial and ethnic identity, just because we want to make sure that we can capture the whole universe of patients that it's in our community. And this is important because while the mutations can be similar, depending on the genetic background of each patient, some of the things can be slightly different. So we want to make sure we can understand that as well. So this is just a very quick overview of how many samples we collected every year. And here we are just sharing the patient samples. We also take samples from parents, and I'm not considering them here because we just wanted to share regarding the patient-specific cell lines. And these are the PBMCs. Now, from the PBMC, which is a fraction or a part of the blood that we collect, we are little by little reprogramming or making them iPSCs. We don't have the bandwidth to make everything at once, so we are prioritizing the cell lines. But I am extremely happy to share that even now I think that I made this chart in February. We had 6 reprogrammed cell lines. Now we have 9 different iPSC cells from patients that are KAT6A, KAT6B, and we are currently reprogramming more cells. Sadly, some of these things are not successful. That means that we have the blood cells, we store it here, and when we are ready to make them induced pluripotent stem cells, we saw them. And some of these lines don't do well. They just didn't survive, or they are not being able to be reprogrammed. For this reason, it's super important that if some families want to contribute again to the bank, they can feel that they can donate the blood, and we're going to store it here safely. So to guarantee that we can have most of the patient samples here. The core facility here at the CREM is now in charge of reprogramming these cell lines and also distributing them. The reason of why we passed through this from our lab to the core is because they have a well-established system to do this in a faster manner. So I don't become a bottleneck in answering emails and allowing the free sharing of these samples. So this is what I mean. The CREM, this center, dedicates the whole facility, the whole establishment to do this since 2013. And this is just a warm-up just to show that since the foundation of the center up to now, we've distributed around the globe, not only these specific cell lines, but also reagents and protocols and things. So this map is just showing how many cell lines we distributed around, not only in the US, but also in different parts of the world. So we're going to be putting the KAT6 iPSC cells into this pipeline. Now, I will have requests about sharing PBMCs or the blood of the patients. We don't share that just because we need all of those samples to actually make the iPSCs. I need everything, all the blood that we have from each patient to give the better chance to have stem cells. That's the reason why we don't share the PBMCs. But I think that now the foundation is doing something, an incredible effort to be able to also share these kind of samples in this conference. I think that, Natasha, correct me if I'm wrong, I think that there is also an effort to collect PBMCs for distributing. Is that correct? Okay, we can check on Natasha later. So the other thing that was also important for us, oh, sorry, one more thing. If you want to participate in this study, there is this QR code. I will also paste a bunch of links about many of the things I've been discussing now, so everybody can have access to all these links without having to pull out your cell phones and scan this QR code. So I've been talking about PBMC and removing blood from patients, but what does actually mean? And now I want to actually introduce a second study for which we are recruiting now. And this means that if you already donated samples for the iPSC bank and you would like to participate in this study, please use the same QR code and sign up again. And I ask in that form, have you already donated blood and would you like to participate again? You say yes, and that means that you're going to become part of this study. And we're going to be at the conference signing this consent just to explain how we take care of the samples, of the information, how much we protect every participant of the study, and also to share a bit more of what this study means. So all the questions we can answer there. So when while we just see blood when we have some blood draw, in that sample we have several cell types, and we basically for the reprogramming, for the iPSCs, and for this new research study that I'm going to share with you in the next slide, we use that part that is called PBMCs, which are the peripheral mononucleated cells, and basically are cells that have genetic information that can be reprogrammed as stem cells, and that we can also use to query to understand a bit more of some health conditions of the different patients. So what we do with these cells, we isolate this specific fraction of the cells, and we remove all the rest, and then we grow them for a few minutes, and we add tags that have different colors. And this is super important because now we have the ability to ask the question of how many different cells we have in the same sample, and what's the status of that cell. And with status, I mean, is that cell healthy? Is that cell thriving? Is that cell reacting to a stimulus? And how does the epigenetic landscape of that cell look like? And with this, I mean, how many of these marks we have, where are they, and are they affected when we have a pathogenic variant in one of the KAT6 genes? And this is something that might sound very standard, but it's actually, this is the first time that we can do this with this level of resolution. So from one blood sample from a patient, we can have a huge amount of information about not only the cells itself, the immune system, but also about the epigenetic status of those cells. And this is something that we established in the lab, and we think that we can. The first question we want to ask with this technology is, can we capture a fingerprints from patients just from these blood samples, and can we use this information to test the efficiency of different drug treatments, or even the evolution over time? Can we use this information to understand a bit better what's going to happen with the evolution of some of the clinical manifestations that patients have? So we believe that there is a strong path with this technology. So this is just a quick example. What does this look like here? I'm just showing blobs of cells. Each of these dots is a cell. PBMC is that fraction of those group of cells from blood, and we can have this technology telling us from all those cells that in our naked eyes is very hard, from a very expert blood expert person, they can actually tell you how many of its cells you have, but this level of resolution in several patient samples is very hard to achieve. So we can do it with this technology. And then apart from knowing what kind of type of cell we have, we also ask to these different software and processes that we use to analyze this data, is there any pattern arising from these groups of cells? So one cell type that was all, let's say, red in this case, we can have it in different colors. And now you may be asking, well, what's separating these cells? Why are you picking these differences in these cell types? And that is that now we can measure what I share with you, this histone modification, the way that the information is accessible to each of these cells. And we can say in sample one, which could be one of the parents donating this blood, you will have this color combination with the specific height of each of these peaks. And then in sample two, which would be a patient sample, their son or their daughter, you can see how different these peaks and the height of these peaks is from the patient to the parents. And this is what we call signature. This is what would be the signature or the fingerprint of pathogenic variants of KAT6A. And you can imagine that if we are suddenly moving, I'm having my finger close, my fingers crossed here, when we have the ability now to test through clinical trials the efficiency of a specific mitochondrial cocktail or drug, or something that we know that is making a positive change in the quality of life of our patients, then this could be one of the measurements that we can use to give the data that is necessary for these clinical trials to move forward. So this study that we are starting right now is actually to be able to collect as many samples as we can from our patient community and ask the question of, do we have a specific signature that is driven by a pathogenic variance in KAT6A and KAT6B? And then can we use this to understand which ones are common across patients, which ones are different across patients, and do they change when we subject them to different drug treatments? And with that, I'm gonna say thank you and take any questions.
Hi, Angie. Thank you very much for that. I was curious about, did you have an explanation for the pretty striking sex skew in KAT6A versus KAT6B? That's really unusual. It's not, that's just not sampling error. That means something biological, in my opinion.
You mean when collecting the samples?
Well, I can't, it was which one was mostly male? KAT6B was mostly male. If you go back to one of the first slides, and KAT6B was mostly female, or it was about even. Yes, there. Right. So the sex distribution, the females, it's about equal between KAT6A and KAT6B, whereas male, it's really, that's much more than one would expect by chance. In a disorder like Smith-Lemli-Opitz syndrome, with which I worked, at least in the early years, there was a strong skew for males because they had genital abnormalities. They had hypospadias, for example, that was not seen in females. So the males are much more easily identified clinically. But when you're these days, when most of the patients are identified by sequencing, one wouldn't expect that kind of this skew in the sex distribution unless there were, for example, increased female lethality before birth, and that happens in Smith-Lemli-Opitz. But I'm just saying that difference, it could be. How many total KAT6A do you have? Or I mean, KAT6A and KAT6B?
So we have around, I should actually have the hard numbers instead of this plot. We may have around 16, 17 KAT6A and then 13 KAT6B, both sexes.
It perhaps that might be jumping the gun. It's even for a small number, that's a greater than 2-to-1 difference. So it does, as I say, when most diagnoses are made genetically, one would not expect a 2-to-1 difference in ascertainment unless there were an effect, a survival advantage of being male for KAT6A, that the malformation, for example, the malformations in females will be more severe in the females, but but actually that if it's any in other syndromes, it's the opposite. It tends to be the females that do better, not the males that survive pregnancy. So it's I guess it is, if one did statistics, maybe be at the edge of significance. Maybe it's as I say, is jumping the gun, but that really struck me as curious.
I appreciate your question, Dr. Kelly, because we actually track this information in case these kind of observations arise, just to understand how many samples we're collecting. Is there any future correlation? In the future, we do want to use all of this information to support the the disease prognosis that I was sharing previously with these epigenetic signatures. But thank you for highlighting that. I'll keep a close eye into that. I was actually, so all this information, it's live in the KAT6 biobank status that the foundation shared a few days ago. So I'm just checking right now into the most updated plots. No, it's still holding like more males than females. I'll keep a close eye now to that, Dr. Gay. It's very good observation. Absolutely. I have a couple of questions. Okay, thank you for your informative talk. I would like to know which cell line is the best for studying KAT6. Which cell line do you work on in your lab? Yeah, I have very strong opinions regarding which cell line is the best. I think that all cell lines are the best. It's very hard for me to establish an order of priority towards deciding which cell line we're going to do start studying. So what we do, I can share what's the approach that we have. So we collect all the information that is available. We first make sure that we have a genetic report. This is important for the whole scientific community. So there is like a genetic report saying where this pathogenic variant is located. Is there any other pathogenic variant in a different gene? Or even if it's, there is this famous VUS, variant of unknown significance, which we can discuss in a different probably meeting or when we get together in the conference. But basically that means that we don't have enough information to know whether that's causing some of the clinical manifestation that patients have. So with all that information, the questions that we ask is, are these clinical manifestations shared across variants that are located in different places of the gene? Is this information, this clinical manifestation or this strength of disease in terms of how much is affecting the development of the children, the speech impairment or congenital heart disease, is that shared KAT6A and KAT6B as well? With all that information, then we say, okay, these are the cell lines that are most likely to benefit most of the patient community. But I do have to say that I took it as a personal / professional mission to, once that those cell lines are differentiated, we are also moving forward to make an iPSC with other variants that some people might think, well, they are not that informative, they are a bit more unique, but we definitely want to make sure that we can, as long as we have resources and funds, we're going to move forward to the best of our ability with all the cell lines. If that doesn't happen in the lifetime of this lab, because we never know what can happen with the current funding scenario for most research programs, we do that. That's one of the reasons why we moved all the bank to the CREM, to establishment that has years of tradition doing this. So the donation that you do to this research program will last longer than any research lab in particular. It's a resource for the KAT6 community. So I hope that that answered your question. Then I think that Mike reached the recording limit, and no questions over there. I don't know, is there any other? Because it carries the most mutations in patients. I don't know. Probably Dr. Kelly could answer this question better. From the samples that we have, I think I shared, we do have that question quite often. So we want to know how that looks like in our hands with the samples that we have in the bank. This is actually the webpage that I was sharing with you. And this is like, I update it very often. Well, with very often, I would say like every 2 months. I update this information. So one of the things that I have here is actually kind of towards that question that you asked, if there is any specific domain that has more mutations or not. So I don't know. Well, in both genes, we have a huge density of mutation in this acidic domain. I don't know if this is representative from the whole patient population, and Dr. Kelly has a huge expertise and tradition studying both of these mutations. I don't know, Dr. Kelly, if you think that this is something that will hold through across the community, or if this is just the quality of our bank having more of these pathogenic variants in the acidic domain.
Most of the mutations are in the second half of the gene, and most of the second half of the gene is in KAT6A, let's say, is exon 17 or 16 and 17. And those are the more severe mutations generally occur later in the gene due to this phenomenon called dominant negative or nonsense mediated decay, that the patients with mutations later in the gene are more likely the protein that is made is more likely to interfere with the function of the normal protein than mutations occurring earlier in the gene where by various mechanisms the RNA is lost and there's no protein made. And that's certainly what I see clinically and biochemically, that it's less severe for the mutations in the proximal portion of the gene for both KAT6A and KAT6B. From a biochemical standpoint, KAT6B overall is less severe than KAT6A, but they have just as many congenital malformations. Both syndromes have just as many congenital malformations of different types or groups of malformations, and so that what I'm saying that the difference between the two is mostly not the biochemistry of the two different genes, but the tissue distribution, the effect in different tissues appears to be the difference. But yes, I saw the distribution that you have. So you have mutations across the whole gene. So one can ask some of those questions. Obviously, I can provide for most of the patients you have, probably I can provide details to say how one could classify the severity or if individual organ systems are involved. Like there's a subset where they are more likely to have bone marrow problems, another subset where they're more likely to have mitochondrial dysfunction. And then there's some overlap. So eventually the interest will be, for example, to get a hematology laboratory interested in the hematologic problems, which are the abnormalities in the bloodlines is from a deficiency of methionine, and some patients have more of a problem than others. So that could be studied. There are lots of things to be studied. And for example, again, with hematologic abnormalities, the question is exactly why is methionine, why do patients with KAT6A and KAT6B need more methionine? Why can they develop clinical signs of a deficiency of methionine even though they're on a normal diet? We see this in other mitochondrial problems, especially more biochemically more severe mitochondrial diseases. It's unusual to see this much bone marrow dysfunction in what I would consider, from a metabolic standpoint, a relatively mild degree of mitochondrial dysfunction.
Thank you, Dr. Kelly. So to quickly summarize the response to Hariso, and please let me know, Hariso, if I'm not pronouncing your name properly. Dr. Kelly's information just shared does align what we have in the bank right now. In the second half of the gene, we do have most of this in the acidic domain. I don't remember right now if the exon 16, I think exon 16, 18 are a bit farther down, close to the serine methionine domain, but I don't remember very well. But it does align with what Dr. Kelly just said. Regarding the blood lineages, Dr. Kelly, we are actually testing some, I didn't give too many details about this right now, but in this PBMC panel, we're testing some mitochondrial markers as well, just to follow up in what you shared. very mixed domain as domains conserved domain acetyltransferase. That's correct. How the different color code per histone is generated measuring blood cells. I am very surprised that very small found in the mixed region. So it's not that a very small number of mutations was found in the mixed region. This is what we have in the bank. So that doesn't mean, while we do have a large collection of patients, that doesn't mean that we have all the patients. So these plots are not representing anything specific about the dynamic of KAT6 variants in terms of the location of the gene. It's just showing what we have in the bank. That's why for me it was very important to ask Dr. Kelly, who has more experience seeing different patients, whether these data align with his knowledge. Regarding the colors of the cells, what we do, it's something, we didn't reinvent the wheel. We just use it, instead of using it for a car that can fit 4 people, we're using it for a huge bus full of people. So what we did basically is to learn from different fields. One of those was the blood field, and use the technique that they use. There are antibodies or little, the same antibodies that our immune system has. In research, we use them to recognize specific parts of a cell that gives them the identity. For example, we know that all cells of one group will express, let's say, or will have an A in the surface. So we have an antibody or a molecule that will recognize that A, and then in there these things are commercially available. That's why I think that this has lots of potential. There is something that has fluorescence. And with this, I mean the whole spectrum of light that has been designed for many of the companies and industry. There are up to 40 different colors that have been generated that are compatible to all be read in the same experiment. So in one experiment, we can fit 96 samples. That might sound trivial, but it's a huge amount of sample for one experiment that is worth, I don't know, 10 samples would be 1,500. And then when you want to do some other high throughput or some other kind of technology like that, it's like $10,000. So this is affordable for most research labs, which is something that really encourages me a lot to make it accessible to many research labs. So we can identify those things are designed already, and you can purchase them. If they are not available, there are different commercial products that allows you to generate the combination of specific color with another antibody that will recognize a different cell lineage. And then the combination of that is read into something that is called spectral flow cytometry. And that allows us to see the whole panel of 40 color if needed. Now, what we did is we modify this thing in several ways. The first thing is that we can do it in fixed samples. That means that, let's say there is a biopsy, or there is a sample coming from a different surgery, or something that was stored in a freezer for many, many years. We can take that and use it to get this information with high accuracy. So that opens the door a huge amount for rare disorders in which the number of cells is it's very limited, and sometimes we don't have the time to process samples. So that's the first modification we did. We can use this in samples that are fixed. The second modification is that we identify a way to get into the nucleus of the cell and query all these histone marks all together. And we validated this, so we know that they are not getting in the way of each other. So the technology is sensitive, and it's also quite accurate. And then the other thing that we also did is, like, nobody in the field knows how you have all this data now. How do you make sense of that? And for us, it's very important not only to generate the resource, but also to allow everybody else to generate it and to understand the data. So we developed a pipeline that allows any researcher just to access the code and just analyze the data themselves. Right? For us, it's super important that everything we do is reproducible and it can be applied for any lab around the world. So those are the differences that we add into this protocol. That's overexpression. Yeah, so we, so Harissa, you're asking about overexpression of KAT6 in the clinical manifestation. Stephen, we are not doing overexpression experiments. In our lab, what we are doing right now is, and this is specifically with KAT6A, and now we want to move forward. We are trying to get the funds to do it with KAT6B. We are trying to control the dosage of KAT6A in the cell, but that wouldn't be overexpressing. Would be if we have 100% of the protein, this is what we see in the epigenetic landscape. If we have 80%, or if we have 70%, 40%, how things change, and that will give us the opportunity to identify specifically what are the direct effects of the amount of this KAT6 protein. And I don't see more questions in the chat. I don't know if there is any other questions.
Thank you. Thank you very much, Dr. Serrano. This was very helpful.
If there are any remaining question or anything, please feel free. I might take a few days to respond, but I will definitely respond. I promise you. I need to go over the 1,000 emails, but I do prioritize if you say KAT6 question or patients, I will make sure I can go through your email first. And also, I'll generate a Q&A question that you can just sign up whenever you can. Make myself available through my schedule, so you can sign up and just ask questions. And it doesn't have to be a huge amount of people. It can be just a one-on-one. For me, it's super important that there are no questions that have not been answered in the best of my ability.
And thank you everyone for attending. This will be available on the YouTube channel of the foundation. You know, if you want to look back, look up the slides, we'll download it on the KAT6 Foundation YouTube channel.
Natasha, I also have a document with the links of everything that I talked or discussed here, and I'm also happy to share the slides. I can give you that, and then you can make them available if anybody wants
Sure,
them.
yeah. If you email it to me, I can put it on the family support groups too.
Wonderful. Thank you so much, Natasha, and thank you everyone for
Thank you,
asking questions.
everyone. Have a good night.
Bye bye. Have a good night.
Bye bye. Bye.